view of what my interests are scientifically. And so I'm directing the Institute for Computational Physics, and what we do there is trying to address complex fluids or soft matter by means of computer simulations and also, let's say, simple theoretical approaches. So our research can be divided, let's say, threefold in parts. One, we develop computational methods and algorithms by which we can then study systems, and there we have developed a parallel molecular dynamics package. It's called Espresso, which we use very much in our research. And Espresso doesn't stand for the coffee, but for an extensible simulation package for research on soft matter, and that leads over to what our interest is. It's actually soft matter. And what this package can do very well for you is to treat long-range interactions very fast, because long-range interactions, because they are long-range, they take normally up quite an amount of computer time, and you are happy if you have developed algorithms which can speed up that part for you. And so we have done some algorithmic developments for just plain Coulomb interactions, but also, and I come to that later, in areas where you have dielectric interfaces, we have, let's say, very fast iterative methods to treat these things. And then one also nice thing is that we have a parallel package working, which is a mesoscopic fluid method, which is called the Lattice-Boltzmann algorithm for coarse-grained hydrodynamics. So you can couple that to the moving particles. I will show some examples of that later in the talk. And we also have recently done an iterative Poisson-Boltzmann solver based on finite elements, which can also do quite some complicated boundary conditions. So all these methods are then applied to biophysical problems, like they are listed out here. I don't read them out to you, and some soft matter problems. And today, because I can't possibly talk about all of that, I will just handle these three things here: macromolecular transport, which runs into DNA separation techniques once you go to charged macromolecules, and this deals basically with charged polymers and also charged colloids, but I will just very briefly touch the charged colloids. When you think about the physics of these problems, you have to think about what are the interesting time and length scales of your system, because that will later on make the choice for you what is the most appropriate tool by which you can investigate the system. And the soft matter area lies mostly in this area here in the middle, where the important interaction energies are actually of the order of kT. And that makes soft matter so soft, because via thermal fluctuations you can induce phase transitions, and their thermal fluctuations turn out to be really crucial for functioning also of biological systems. And so means you want to study this system, but what kind of input do you have to have? Well, if you're interested in the really detailed interactions between these entities, then you probably have to come from the left-hand side, starting from, let's say, some quantum mechanical calculations. But there you are on very short length scales, very short time scales. That means you will always be limited in what kind of system sizes you can address. And therefore, what people try to do is to always get some information out of the quantum area, maybe transfer that to what's called the atomistic approach, where you now resolve your system, let's say, down to point atoms which interact via some effective interactions, but that normally is still too small, although with the advance of computational power, people can now, let's say, address soft matter problems on a small scale already atomistically, but still the length scales which you can address are of the order of 10 to 15 nanometer cubed, or let's say 20 nanometers if you have bigger computers. But this is very often not enough to look at macromolecular transport of DNA strands, which are, let's say, micrometers in length, or look at membranes, which also cover much larger areas. And then, of course, on the right-hand side, what you still want to do is to maybe make contact with continuum theories where PDAs govern the behavior, and so there's this interesting idea of coming up with multi-scaling approach where you successively take into account input from lower level or higher accurate theories and then try to push that to the limit of, let's say, continuum theories. The theoretical methods which we are going to use are of course different depending on which part of the problem you're looking at, and so means on the I mean what we are basically trying to use or using are here on the meso and micro scale. So it starts out with dissipative particle dynamics, which is some tool for hydrodynamics, hybrid MDMC, Bettis-Boltzmann approaches, and on the lower scale we do classical force field simulations where we really resolve these systems on the atomistic scale. So anything between here and there we have some knowledge and we try actually to link these scales, but one should be aware that of course there's still the macro scale and the very quantum scale which one eventually also has to bridge. I don't know what happened here, sorry. It's back, yeah, but it was somehow shaky. Here we're back. So let's talk about the topic of today, which are these charged macromolecules. And so let's look why are charged macromolecules interesting. What are charged macromolecules? That are those macromolecules which dissociate charges in aqueous environments. So on the atomistic level, you throw some salt in water, it dissolves. It gives you ions, pluses and minuses. If you have a macromolecule, which is like a polysalt, you throw it in water, it can also dissociate ions. And some of these synthetic examples of polymers are these polyacrylic acids, which have a dissociable sulfonated group here. So that gives you a sodium ion, which swims in the solution, and the backbone of the polymer remains with a charge. And because polymers have many of these macromolecular units, you have a long string of a polymer which is charged. The same thing happens also in biology, because that's why charged macromolecules are of prime interest in biological applications, because basically all of our biological systems carry in one way or other dissociable groups. So the cell membranes are sometimes charged, DNA and RNA and proteins carry dissociable groups which are, to complicate things, pH dependent. And on the application side, I mean, these charged polymers are very interesting for applications like in superabsorbers. So you might have gone across that at early stages of your life. In basically every diaper there are these polyacrylic acids which absorb a huge amount of water, and it's actually a $1 billion business, Procter & Gamble for instance, but they also used in paper industry as viscosity modifiers, additives to detergent to avoid calcination and waste management. So there's plenty of applications of charged macromolecules. Well, in biology already said, I mean, gene transfer mechanisms depend also on how viral vectors can go through the cell membrane electrostatic barrier, understanding biological functions like in ion channels, and also the case of DNA packing in viruses are of interest where people are looking currently at. So there's also a large tendency to self-assemble in nanostructures. I mean, this is clear. If you have charged substances of opposite charge, I mean, they will find themselves and assemble very easily. Give you just some nice pictorial examples. This is a hydrogel, huge macromolecule contained probably of little granulars. This contains almost 90% water, and you can still hold it in your hand and it doesn't dry out. So this is a good example of how nice a super absorbent material actually can look like, and what you can do with it. These nanostructures can also assemble even in like charged objects. So here on the right hand side you see a ring which is formed out of DNA together in some special salt solution, which makes actually the DNA, which is negatively charged and actually a high persistent length, means it is like this rod here. I mean, it needs some energy to bend it, and it still forms these toroidal objects. This is due to some charge correlation effects I'm probably not going to talk about, if you take some synthetic polymers here on the left-hand side, which are also charged, they also form some filaments, and like here, these actin filaments on the right-hand side, biological substances, on the left-hand side, synthetic polymers. So there's a generic mechanism of forming structures due to charge interactions. And the second example, which looks nice, is here this T5 bacteriophage, which again is some DNA which comes out of this virus, where it was highly compressed. It was released through this funnel into a vesicle, and still you see that when it is released into this vesicle, it forms this toroidal shape, which we have seen already on the other side. Now, but this is all equilibrium physics, so still interesting, but not in the interest of what I'm trying to do here. What happens if we apply a field? Well, this can lead, first of all, to non-equilibrium phenomena, to interesting phases, like there are known stripe phases in solutions of positive and negative charged colloids, for instance. It's biologically relevant because we have macromolecular transport, which is driven sometimes through potential difference, like in this nuclear pore complex or in ion channels. You can manipulate charged macromolecules through external fields. Structure and dynamics investigations are normally done also by applying electric fields, here AC fields and dielectric spectroscopy. And the last point, which I'm going to talk a little bit more in detail, is about electrophoresis. Basically DNA separation techniques where you apply a field and you try to separate, for instance, DNA by their length because it might move differently according to an applied field. So why is electrophoresis interesting? Well, as already said, one is interested in separation of DNA because nowadays it has become customary that we want to analyze the genome, but you want to analyze it very cheaply. Mostly still people use gel electrophoresis based on entanglement, but this is very slow and therefore also expensive. And the future are basically novel separation techniques which are based on hydrodynamic and chemical interactions. And so this is an ongoing development process, and I will come to a very fancy way later on in terms of these nanopores. But our first goal, which I would like to present to you here, is to understand free flow electrophoresis, and I would like to show you what kind of methods you can use there. In free flow electrophoresis, as I said, this is the pictorial description of a polyelectrolyte, so a charged polymer with the counter ions here in the solution. You apply an electric field. The polymer would like to move in one way according to the applied electric field. It experiences, though, a drag in the fluid, which is a force into the opposite direction. Also, the counter ions would like to move into the opposite direction than the polymer. And it is not an easy problem to solve this analytically, because you do not know beforehand what is the correct charge which you have to apply, because the electric driving force is something like Q effective times the electric field, and the Q effective is something which comes through the coupling of the counter ions towards the polyelectrolyte, which again is a multi-body effect because it depends on what kind of concentration you are, how much is the background salt, what are the conformational degrees of freedom of the polymer. So that is analytically basically impossible to handle, and that's why computer simulations are for this kind of field very good tool. And to get out the electrophoretic mobility, what you do is you equate basically the electrostatic driving force, you equate the drag force which acts on this object times an effective friction constant times the velocity under which this macromolecule moves. And then because in equilibrium or in transient state they have to be equal, that leads to the quotient here to the electrophoretic mobility. And if, let's say, if you just plug in these equations, you come up to Q effective over gamma effective, which can depend on the degree of monomerization of your polymer. So depending on how long your polymer are, these values will change. And for DNA, it turns actually out that for very long n, gamma effective scales exactly, namely linear with n like gamma effective, and you can't separate really long DNA. What means long DNA? Well, that's
how realistic free flow assumption?
Sorry?
How realistic and biology free flow assumption?
in biology. So, I mean, how realistic is the free flow? It depends on how you set it up. I mean, I can set it up that I have a free flow. I mean, I have just the DNA in solution.
Yeah, but that motion of charges and polymer flow produced by this motion.
Yes, it will produce some effects on hydrodynamic nature, which you would like to take into account. And we have a method of doing that.
Mhm.
Actually, the mobilities are very slow, so you're definitely in the very low Reynolds number regime, so there shouldn't be anything drastically happening unless you look for nonlinear effects. Maybe it becomes clearer later, otherwise we can discuss again. So an experimental observation of the mobility basically looks like this. If you have very short fragments of DNA or any other polyelectrolyte, and so I'm moving away now from DNA because our experiments which we compare with are not done on DNA but on sulfonated polystyrene. So you get them in these different lengths. You can measure the mobility and it goes somehow linearly up, it goes through maximum, and then you reach what I was calling before this free draining limit. Namely, then the mobility does not become length dependent, and this free draining limit, so this crossover happens for DNA already around 170 base pairs, and the base pair is a separation of about 1.7 angstrom, so you can calculate that it's not very long, and for PSS it appears about 100 units. So that is what people observe, and what one understands is for this limit there is theory that this should happen actually. This linearly rising part one can understand also on basis of let's say simple theoretical arguments, but why there's a maximum, this was basically not quite clear, and that was our point where we said okay, that's what we would like to understand. Now coming to the model, what kind of model do we apply? Well, you can't really treat the system atomistically because once you go to say more than 10 monomeric units, atomistically you couldn't handle this on the computer anymore. So you use what's called like a bead spring model for polymers. So each of these polymeric units get mapped onto one bead. Because it's a charged polymer, you also have a charge in this bead. The counter ion again is also just a bead with a charge. The water is completely coarse-grained away into this implicit solvent, which you describe by its relative dielectric constant, and that temperature, which is 80 for water. And then, if you coarse-grain the water away, I mean, you have, of course, first of all, no choice of looking at hydrodynamic interactions because it's just an inert background. But there's a trick by coupling now all these beads to what I was alluding to before, to this lattice-Boltzmann solvent, which is a coarse-grained description of some solvent which retains the hydrodynamic interactions, and that is what we're doing here. And I will.
So, excuse me, Christian.
Yeah.
What are the black solid dots? I see something like a filament there with some minus charges, but what are those black ones? Yeah.
Okay, in for instance PSS, not every monomeric unit is charged. So therefore these black dots are again just beads with excluded volume, so just spheres like marbles, but they're not charged. But they can interact with the other marbles via the string. Entropically, it's called an entropic interaction because that gives the polymer its entropy, and you have kept the excluded volume interaction. If you're very ambitious, you can also include in these beads some more effective interactions of short-range nature, but for us it's a coarse-grained model, so you're happy. Let's think about just having black marbles. The color is actually irrelevant, they are just hard. They are just interacting via excluded volume.
So I imagine that the positive ions on the outside are completely screening any kind of interaction from loop to loop.
So that depends on the concentration, because what goes always in there is of course entropy. You are at ambient room temperature and you have a big bucket of water, so the ions actually, I mean, they are pretty happy if they are, some of them, if they are far away from the chain due to entropic degrees of freedom, and you have a competition with the electrostatic field. So therefore, and you have thermal fluctuation, so the ions, they are wiggling around. There's a density distribution which you can calculate in some limits how the ion distribution should look away from the polymer. And that is all taken care of basically automatically in the simulations, because you put all these assumptions in your
What about
algorithm.
the polarizability of water? If you use water for solvent, right?
Yep.
And it should screen the charges interactions.
You have just dielectric screening with this model.
Yeah, right.
So it's an epsilon r, so the dielectric relative constant of 80. So the Coulomb interactions are reduced by a factor of 80, so which is this dielectric screening of water. And then you have point charge interactions. Well, point charge in the sense that, of course, these are not point charges, but they have an excluded volume interaction. This is also to prevent the Coulomb catastrophe. I mean, two ions cannot run into their centers, because then you would have a divergence in the electric field. That's why you give them actually an excluded volume, which is also true in reality. I mean, ions have a size.
So why are you thinking about each of these circles as just a charge and not something with structure like a frame that has, a few sticks, that have charges on the end and have something to do with the orientation of this frame? Are you thinking about that?
You could do that. I mean, if you are interested in making a further refinement, I mean, that's basically what an atomistic model does. An atomistic model, instead of having just one bead, you have, 6 carbons around in order to make, let's say, the benzene ring, and then you give partial charges onto these carbons, and you have some more dihedral interactions. I mean, it becomes more and more complicated. You can do that, but then the amount of computation time it takes grows, and so you don't want to do that for polymers, because polymers, you want to make them very large, and that you can't handle by making the interactions too fine-grained.
So let's talk. I have a theory which allows for the little frames along the polymer.
Okay, can discuss that later. Yes. So here's this basic idea of the simulation which we do. I mean, it's an MD simulation, in particular ensemble is NVT. So you solve Newton's equation of motion. You take a thermostat which gives you some noise into the system, which you take out by friction. You have very simple interaction potential like electrostatics, hardcore, and I didn't put in here the polymer degrees of freedom. You use periodic boundary conditions, because your system is effectively infinite, and you run it with your program of your choice. The hydrodynamic via this lattice-Boltzmann fluid works now that you have sort of a lattice-Boltzmann equation on a lattice. You have two steps here, a collision and a streaming step, and this fluid is actually a very simple one, because it just contains a discrete set of velocities, and the discrete set is actually quite small, because you have 19 populations which you put on this three-dimensional lattice, and still this is sufficient via this discrete Boltzmann equation to generate you correct hydrodynamics, well, in the limit of infinite of zero lattice spacing, of course. But now what you do is you have a background fluid which can move, so to speak, on these lattice nodes, but it's also coupled to a particle which moves in the continuum. You do that by basically making a coupling between the particle degrees of freedom with the lattice, and this coupling is basically a modification of Newton's equation, where you look at the velocity which resides at a specific node in the lattice-Boltzmann fluid, and you take the difference to the particle fluid, and then you basically apply a frictional coefficient by which you can exchange now momentum between the particle and the fluid. So the fluid can push the particle, transfer some momentum, in the same way the particle can take the fluid along and transfer some of its momentum to the fluid, but all under the condition that the total momentum is conserved. And in this way, you have a true description of the hydrodynamic interactions. So this is coupling was invented by Ulrichs and Dinweg, and we have it implemented with actually thermal fluctuations, actually with the correct fluctuation spectrum in Espresso, and we also have a GPU version of this available in Espresso, which makes this algorithm actually very competitive, because now it is as fast as the molecular dynamics in the background.
So Christian, is that formula about the velocity u sub I of the fluid parcels? Or is it, yeah, what is u sub I?
U sub I, let me go back.
V sub I, what is V sub I?
V of I is the big particle, I mean the particles which move in the continuum. This is the velocity of the particles, and the U is the velocity of the fluids.
And so separately you're calculating the ULB.
Yes,
Right.
because that, in principle it leaves completely separate from the particles in principle, but we have coupled it. I mean, you have one layer where you have just the lattice fluid, you have another layer where you have the particles which move in continuous space according to Newton's equation, and at each time step you actually look where the particle is, you look what is the velocity around this particle, and you have this coupling between the relative velocities, and if there's a relative velocity, then you apply a drag force.
And boundary condition on the surface of a particle?
It's a frictional coupling, so you don't have
V
to
not equal to...
you don't have a no-slip boundary condition, because your particle is a point particle. It's a point particle.
Point like that.
Yes, which
But this was only because it is a sphere. If it were elongated, you might also consider the balanced boundary.
yes, we are working actually, there's a different kind of coupling which is called the lab coupling, where you would like to implement some, let's say, no-slip boundary condition of a larger colloidal surface immersed in this approach. You don't have to, because there's always a way around that you say, okay, every more complicated shape I can assemble like in a Lego fashion with point particles. So if I want to have a big colloidal sphere which has also rotational degrees of freedom, then I just make it up of little beads, which are basically the interaction centers which interact with the fluid. And then I can have rotational degrees of freedom, also translational degrees of freedom. So that is one possibility, which at some point gets costly and has some other problems, but there are other boundary conditions you can think of. Right now it's just this frictional coupling to these point particles.
So where are the filaments that you started with?
The filaments are made out of these beads. They are still floating around. I haven't showed you to them. I mean, I will show them to you later. Is that okay? I mean, here's just, this is just a teaser. I mean, this is not the real system we are discussing. I mean, this is just basically to show you that with this sort of hydrodynamical method, we have particles coupled to this invisible background solvent, which you don't see. We let it run by a pressure-driven flow through this obstacle, and you have this rolls developing. And of course, I didn't tell you, I mean, how much this computation costs, but actually on the GPU you can almost produce it in real time. It might be a factor of 10 slower than this, but it's really fast. But it's not the system I'm studying, so you don't see filaments.
Actually, it looked like the simulation stopped just when it started getting interesting.
Yes, that's always the case. Let me continue coming to the filaments. So now we have these particles. We'd like to extract the electrophoretic motion. So how do we do that? Well, there's actually some other thing which are experimentally interesting. It's just the polymer diffusion, which you can get from this Rey-Kubo relation. You can actually also get the electrophoretic mobility via applying an electric field and observing how fast the object moves. But there's also a way of getting actually the zero field mobility via Rey-Kubo relation, meaning that you don't apply an electric field, but you just look at the equilibrium fluctuations of these charged entities in this way, and this gives you equally the linear response transport coefficient, namely electrophoretic mobility, and that is what we're actually.
That's an equilibrium result, the green Kubo.
Yes, but it's in linear response theory. It gives you the transport coefficient, like the diffusion, electrophoretic mobility, conductivity, and so on. That works.
The formula for the cell diffusion is very general one. It was not derived by Einstein, but it is not something dependent on equilibrium. It's always true if you have a particular problem. You don't need to have equilibrium.
Okay, let's look at results. So what do you see? Well, here's plotted the dependence on the diffusion of the charged filament now, or polyelectrolyte, as function of its length. This is a simulation with hydrodynamics and without hydrodynamics, because I mean we are in this position that we can actually say, okay, we do not use the lattice Boltzmann, so we turn hydrodynamics off. Then the polymer is just moving according to the Langevin equations, which are known to give you no hydrodynamic effects because it's a very local aeroson. And you see the effects of hydrodynamics because you see that they scale with lengths completely different. I mean, no hydrodynamics gives you the theoretically expected result, this linear dependence on the length, whereas if you take hydrodynamics into account, you get the pink or purple looking curve here. And then we have some experimental pulse field gradient NMR results here, and they agree actually quite nicely with our simulations. So showing you that if you want to make connection to real experimental systems, you should not throw away the hydrodynamic interactions, but they are really important. So this
What are
was
the error bars on the experiments?
where are the error bars?
Yeah.
I can't tell you. I do not think that they are that big. I would think that because they haven't recorded them, they are the range of the symbols, which is the same true for the error bars on our symbols.
And D naught, what is D naught here?
D naught is the mobility of a single monomer. So it was rescaled basically to agree here on D naught, and there was a different normalization on the red one.
Yeah, that's, is this the number of parameters in that fit?
It basically, you have one free parameter, which is you normalize via D naught, and therefore you have to agree on one. Now, the same thing trick with one free parameter you do with electrophoretic mobility. You normalize here actually not with on this side, but you normalize with the free draining mobility, which is the one here in the limit of large n. And what you see here again is our simulation are the red points, and this time, the simulation are the this ones. So you have some discrepancy at the low-lying ends of the shorter polyelectrolytes, but what the simulations still show you that you run through a maximum. Okay, the free draining limit has to come out the same, but you also see, of course, some differences in two types of experiments now. I mean, one again was this pulse fields gradient NMR, and the other one was capillary electrophoresis. So this should be contrasted again to a purely Langevin dynamic simulation, where you can, from a simple theoretical arguments, you know that it should just decrease monotonously with the length of your polymers. So that is something which definitely cannot give you that sort of behavior. And let's say because we throw away a lot of details in the short range interactions, it's also not surprising that your results do not lie top on top for the shorter monomers, but the qualitative behavior and also, let's say, the location where the maximum is not too bad.
So in this inset figure, in the vertical lines, is that the experimental error bar?
No, you can't do actually here experiments, because this means you would be a genius and turn the hydrodynamic interactions in the solvent off during the experiment, which you can't do. except if you do it in vacuum. And so, I mean, this is purely theoretical. So just our simulations with applied field doing Kubo and theoretical, and everything is basically within error bars. But this is irrelevant because it just should demonstrate you that the hydrodynamical interactions in this approach are really important. Just a glimpse over the conformations which you have when you do this is that the polymers, because they still have flexibility, I mean it's a completely flexible chain here, just we have these charge interactions, so in this case all the leads are charged here, and these are typical snapshots. Also, I mean, if you look at the simulation, everything wiggles. I will show you a small movie here, and just to illustrate what sort of conformations one expects if the chain becomes longer and longer. And you see that actually on longer the chain it becomes quite stretched. This is in this case due because we're working at very low polymer concentration, that is almost no added salt, and this means that the electrostatic interactions are basically not screened on short length scales, and that's why the chain stretches. If you want to know how an simulation looks like, I mean this is how these wiggles are within some time period. We just colored one of these counter ions in green so that you have an idea where it moves. And the idea was, is there actually a counter ion exchange between the counter ions on one of these polyelectrolytes to the other? And as you have seen while I was talking, that it was jumping from one to the other, and here it jumped again somewhere else. So because of these thermal fluctuations and the weak electrostatic binding of these counter ions to the backbone, you have an exchange of ions, and that brings me here to the conclusion. Just to show you, this is basically some project we finished 3 years ago. That with our simulation methods you can model this theme of electrophoresis. Hydrodynamic interactions are actually, let's say, very important in the subject because you are not allowed to ignore them, and we are able to reproduce its mobility maximum. We have also some other results there, which in the lack of time I'm not going into details. It's just like an overview of what we have learned in this subject. Now other projects on transport, which I just...
So just a second, what about hydrodynamic screening and shading? What are you saying there?
Why am I saying this? Well, because the idea was, I mean, and I don't have the data here with me in order to convince you. I mean, the question is, why was there a maximum? And the maximum was actually coming out. I mean, if we analyzed in detail the effective friction versus the effective charge, how this scales as a function of the length of the polymer, and you could see actually that they scale differently in different areas of length until you come to this crossover length where you reach the free draining, because then everything becomes linear. Everything scales linearly with n, so this means then this quotient Q effective over gamma effective becomes independent of n. I mean, this is the origin why very long polyelectrolytes cannot be separated due to length. But there was this intermediate part where the effective charge scales differently like the frictional part, and this has something to do that actually the polymer, that the friction in the beginning doesn't rise as strongly as the effective charge, and this we attributed to this sort of windshield effect, because if you put on monomers together and you move them through the fluid, I mean their friction doesn't rise as fast as the effective charge because they can move into the windshield of the other particles, and this has led to this maximal behavior. I mean, actually I didn't
realize it would be that cool. Thank you.
Other projects, I mean, we looked at electrophoresis of global charge neutral objects because, let's say, this is also quite interesting because you think, okay, I apply a field, I need some charged object in order that it moves. But this is actually not true because there are some cool, from my perspective, electrokinetic effects, because this is just the perspective that you just have one object in the solution, which is not true because you always have the counter ions which are surrounding even a globally net neutral object, because there are these dissociable groups, they have dissociated charges, but they are distributed somehow around. Now you apply the field, these mobile ions start to move, they create a flow because they are coupled to the fluid underneath, which is called an electroosmotic flow. If you have positive, negative ions, the flow goes in different directions, and this makes, for instance, an object like this move. So this is, let's call it Ashdari's cross. It's a globally charged neutral object with a very definite charge pattern. Let's say green is positive, red is negative. You apply an electric field in this direction, and due to the electroosmotic flow, which you see here with these flow lines, the cross moves perpendicular to the applied field. So it's not that it moves, it even moves not in the direction of the field. And this is a two-dimensional, I mean, in order to understand this theoretically, you have to confine that basically to 2 dimensions. But this was worked out 15 years ago by Ashdari and Long by analyzing the flow field. And
what if you rotated the cross? Or you...
If I rotated it, I think it would move just, if I rotated 90 degrees, it would just move upwards.
Rotate 90 degrees, then it moves upward. Okay. But 45 degrees.
45, then it would like to move backwards, I think, into this direction in order to move. Because, I mean, the electroosmotic flow will exert a torque on it, and then you have an equilibrium. I mean, you have the equilibrium position, transient position in which it would like to be. It's already almost time to stop. I just want to glance over a few other projects we have finished, because instead of looking at these crosses, which are, let's say, charge pattern objects which are not really found in nature, but in principle you can make them. Things which are found in nature are these charged colloidal objects, so these colloids. Well, these again are, of course, polystyrene colloids, which are made in order to perform experiments on them. So they carry a high charge, and this experimentalist, the Kremer group in Leipzig, is able to measure the electrophoretic mobility on single colloids by putting them in this light tweezer, and don't want to go into these details, but what they can measure again is the electrophoretic mobility. So you have, like you had with these polyelectrolytes, with the polymers, you have now just charged colloids, you apply field, it moves, you measure mobility, and you would like to understand how is the dependence of the mobility on the salt concentration. Because now if you change the salt concentration, you have different effects of screening. And actually, to make it more interesting, they were using mono, di, and trivalent salts in which they immersed them at different concentration. And what can happen, actually, a trivalent salt is a very interesting charge correlation effect, namely that you can revert the mobility of this object. So a charged object which moved according to its net charge to this direction, if you immerse it in a trivalent salt solution, it can actually invert its charge by absorbing more of these trivalent counterions than necessary to compensate its bare charge, and then it starts to move in the other way.
But this particle are finite size. You have to change boundary conditions.
No, you don't have to change any boundary
It's
condition.
point-like particle.
No, it's a finite size object.
So
All this charge business.
Just not necessarily the same velocity at the contact point as the motion of the particle. So in previous equation, it was pure dissipative coupling, frictional coupling.
It's still a frictional coupling. I mean, it's a question now, do we talk about the methodology to simulate it, or do we talk about the real experiment?
I would prefer a real experiment or real life test.
Well, the real life experiment shows that it reverts its mobility. So you have trivalent counterions, and now you would like to understand the real world experiment. You reverted the mobility, and that comes about that, as I said, these counterions, I mean, if you have multivalent ones, they are strongly more strongly coupled to the colloid because you have 3 times stronger interactions with the surface charge, then because they are stronger bound, there's actually a charge correlation effect, which I could explain you on the level of simulations and some theory as well, but I don't, which leads to a charge reversal, and then you basically reverted the charge. The ions are now stuck onto this colloid by pure electrostatic interactions. I mean, this is possible, but actually we could show that this is not sufficient to explain the experimental results because you needed an additional short-range attraction in order to reproduce these experimental results, showing that in nature there might be more than a pure electrostatic coupling of these counter ions towards the surface, which is not so surprising because you have water effects there, of course. There are a bunch of effects which can play a role here, polarization issues and so on and so on. But as a matter of fact, on the basis of these simulations with pure electrostatic coupling plus additional short-range interaction, you can figure out what nature does in addition to Coulomb interaction. And that's basically the outcome, let's say, of this experiment.
What is it doing in addition?
What does it do? Well, it has an additional short-range interaction.
Additional short range.
Yes, we have not resolved what is the cause of this additional short-range interaction because there are many possibilities, and then you have to dig down, get your microscope out, and put in more detailed short-range interaction. Basically, it would mean that you would have to throw back explicit water in there, and maybe if you're bold, even polarizable issues. I mean, quantum mechanics and so on. Then it becomes more complicated, but that was not the point of this exercise. Right now, I mean, that's actually the reason why I'm here in Cambridge, is because this group, some people are here from Ulrich Kaiser's group at Cavendish Lab, where they look at artificial nanopores, where you want to translocate DNA through in order, for instance, to sense base pair resolution of DNA sequencing and maybe other proteins. And so that's why we developed also some algorithmic models for doing that. This is just, again, a teaser movie because that's not how in reality we model this. So this would be something like a single-stranded DNA going through a nanopore hole. The question is, what kind of information do you need to put in order to model this effect correctly, I would say. And what do you need to put in? I mean, the first thing we thought is of looking at the effects of actually dielectric interfaces. So the DNA in this area now is actually very stiff on the area or on the length scale of this nanopore. I mean, these nanopores can be lengths of 8 nanometers or 10 nanometers, and they can be 10, 5, 20 and longer. I mean, depends what kind of pore you take. But the DNA has a persistence length of 50 nanometers. So on the area of the pore throughput here, it might be sufficient to model it just as a stiff rod. Stiff rod with charges on them, which represent the bare charge of the DNA. And then if we would assume still our implicit water model, then you have now a slight problem, because water is clearly defined in the outside in the bulk to have a dielectric constant of 80. It's clearly in the solid state nanopore, it is a value of about 2. It's something in the pore which you don't know, but already there's something that you have a dielectric interface here towards the bulk makes the simulational issue a little bit more tricky, because now in principle what you would need to do is solve Poisson's equation each time you move a little ion around and calculate the force correctly. And we actually got an iterative method which can more or less do that on the fly on the speed which you need in order to make, let's say, 1 million MD steps, because that's how often in principle you need to do that. So what we did now was just poking this DNA into the pore, measure the force which is exerted on this object while it is at certain positions in the pore. You can integrate that, and that gives you basically the free energy of putting now a stiff DNA through the pore from the left, where it is completely outside to the right where it is again completely outside, and you have a so-called translocation barrier. And we are looking at how this translocation barrier depends actually on the salt concentration. If you take these image charge, that means the dielectric contrast, into account on this continuum model or not, and you can see that the difference is basically between the filled and open symbol, that the difference is of the order of 5-6 kT if you have very low salt concentration, but it basically becomes invisible if you are at the level of 100 millimole. So that means in low salt concentration, the dielectric effect is very big and should be definitely taken into account. At high salt concentration, you can probably safely ignore that. And let's say good for the translocation people is that they always work at 1 molar concentration. So there, this dielectric interface is basically negligible.
And how does there's going to be an entropic contribution to that barrier if the thing is less stiff?
Yes, that is seen on the next slide. So that is when you do it with a flexible, total flexible polymer, and we did the same thing. And actually, the nice thing, let's say for us, is that the barrier increases if you go from a stiff one to a flexible one. Because once you have a flexible object inside the pore, you are more likely to experience incidents where the polymer is closer to the pore wall.
Can you just go back so I can compare the scales? So it's about 15
Yes, I can.
kT for that one. And
So that is 10 maximum
so
at
similar.
10 millimole, and here we are at 20, and this is all done also at 10 millimole. So you have factor of 2 if you have a flexible object. So I think I should stop here. So the other atomistic part I just skipped. These guys anyhow. You can't really make much with the conclusion because in order to understand that you should have seen the other slides. I just want to get an advertisement out for our program. I mean, it's freely available, open source software in the GPL, and for these coarse-grained and Lattice-Boltzmann simulation, I think it's quite nice. At the end, I should thank you for listening, and some of my team members who did the electrophoresis, like Sherwin, Owen, Georg. Stefan, who's doing this nanopore, actually was not in this picture, and Kai Grass, who did most of that a few years ago, is already making big bucks in the consulting industry. So thank you very much.
Time for some questions. Very a lot already, but I guess there are more.
DNA just a uniform rod or double-strand DNA, or you taken into account some non-uniformity due to base pair sequence?
The slides which I was showing you for a longer time, this was just a uniform charge rod. I mean, because you ask, I might say that we also did actually 2 editions. I mean, one, because there was some experimental stuff we didn't get out right. We actually reverted now to completely atomistic simulation. So there you have an atomistic resolution. I mean, basically each atom in the DNA is represented by a force center with some potentials, with some partial charges. You have explicit water of some SPCE type, but you can also just do, I think, 20 base pairs is that we do. You have actually an infinite pore now. You close it via a periodic boundary condition. But then you're able to simulate that in all its detail, and then you find out actually, it might be just a coincidence, that actually we were able to reproduce the experiments extremely nicely, and we could figure out what's actually missing in our coarse-grained model towards this more atomistic model. And this has something to do that there are ions. I mean, we're looking here at conductivity. I mean, we're not looking at translocation, actually just ion conductivity, because that's what's interesting from the experimental setup. They just measure conductivity and they look for the blocking of the current due to the insertion of the DNA. I mean, that's actually what you want to have as a signal for detecting base pairs, because there might be a base pair specific sort of blocking. Now, in order to see that, of course, you have to have quite a fine amount of modeling. I mean, that's not what we wanted. We thought, well, just the ion current, that's something which is coarse enough, which we should be able to get out also on a coarser scale. And it turned out that actually you have to distinguish between the ions which are inside the grooves of the double helix, because those are the ones which do not contribute a lot to the current, because they are pretty strongly bound, first of all electrostatically, but also due to some other water mechanism. And so that's something which leads to a position-dependent mobility of your ion, which you somehow have to put into your model. Daryl.
I can see how the persistence length will vary when you increase the charges on the atoms of the polymer, the beads. Have you got some independent method of changing it for an uncharged bead spring model? I was worried about what happens as you change the pH, say, on the comparison between experiment and the numerical simulations. Does in the numerical simulations, what happens to the persistence length as you reduce the charge?
We have changed the persistence length on this one model where we looked at just translocation barrier. I think it might even be in this plot. No, it's not here. Normally, for this kind of system, I think the persistence length does not play, well, plays a big role, but as I said, I mean, DNA is rather persistent. If you go to single-stranded, okay, it's rather flexible. We can, in principle, tune the persistence length to any value we like by adding a bond angle potential to our model, and we've done that in order to see how much of this going up is due to electrostatic interactions, how much is due to bare persistence effect. So that can be done. I mean, we've just done some rudimentary effects because for us the double-stranded DNA was the prime target, and that is so stiff that in the beginning we were happy actually with the rod model. And I think on this translocation area the persistence length probably has here not an influence, though it definitely has some influence on other issues for finding the pore entrance or other effects like that.
Now, with the single-stranded DNA in a real laboratory experiment, to what extent is the persistence length modified by the charge? Is that an important effect, or is it just the intrinsic?
It's mostly, I think, the intrinsic. I think the electrostatic issue, if you change salt concentration, here's some experimentalists, do you have some idea? I don't think you never measured that, but I don't. It's probably in the 10% range. I would guess.
I don't think we have a direct way of measuring persistence. That's really the main problem.
I think the electrostatic contribution to double-stranded DNA is definitely just below 10%. I mean, for single-stranded objects, and depending in what range of electrostatic conditions you are, salt background might vary.
Okay, thank you.
Final question goes to Darren.
In the beginning of your talk, you said that these systems have a large tendency to self-assemble in nanostructures. But how did your talk really demonstrate that?
Not at all.
Okay.
Yeah, it was not assumed to demonstrate that. Actually, it was just, I started out saying about equilibrium structures that I tried to motivate. I mean, the idea why I put this in was to say, why are charge systems interesting? It is not in my, I mean, I looked at actually structures a long time ago, at equilibrium structures, and that has something to do with this overcharging business, for instance. I mean, when you want to associate multivalent counterions on charged substances, then you see actually that you can also make like charged objects to attract. That was in our line of research a while ago, but found it boring by now, because you can have these attractive interaction mediated by the trivalent counterions, which form like a Wigner crystal, like sort of lattices, depending on what kind of interactions you look at, and these lead to attractive interactions. And people got very much excited about that, and was a big peak of research, but by now I think it's quite well understood. And but it can be used, of course, for self-assembly, because whenever you throw in multivalent ions into a solution of charged objects, you have self-assembly to the point where the thing falls out of solution. Then you're probably not happy anymore. But you can play with that a bit, and people try to get things into structure. But I would think that, let's say, applying electric fields is even more promising, because then you can move objects, you can direct them via gradient fields, let's say, to points where you like to have them, and so on. That's, for my taste,
It seems to me that the interaction with hydrodynamics demonstrated that if these things are assembling in nanostructures, it's clear that that the hydrodynamics has something to do with the process of assembly.
Yes, you can, through flows, you can modify how they assemble. That is, for instance, a colleague of mine just looked at the crystal, colloidal crystallization of charged spheres in the absence, in the presence of hydrodynamics. You find that that can play a role, actually a counterintuitive role. Normally you think hydrodynamics will speed up crystallization, and what they found is actually when you have hydrodynamics involved, it slows down the kinetics of the crystallization. And so there are interplays, definitely, and I would say there's still quite a lot to investigate, but that is not quite on my agenda.
Thank you, Christian.